Welcome everyone. My name is Matthew Macauley, and I'm an assistant professor at the University of Alberta. I'm a GlycoNet network investigator, but today I'm going to be here sort of representing the ACS carb division in this joint webinar series. So without further ado, let me introduce our first speaker, which is Professor Mary Cloninger. So Professor Mary Cloninger is a professor from the Department of Chemistry and Biochemistry at Montana State University. And her research is focused on chemical biology, particularly protein-carbohydrate interactions at the cell surface that are important for cell-cell interactions. She's particularly interested in studying the role of galectin-3 carbohydrate binding protein that is overexpressed in many cancers. Her research involves the synthesis and study of synthetic multivalent frameworks such as dendrimers with carbohydrate end groups, and I think we'll see a little bit about that today. So Professor Cloninger obtained her PhD in organic chemistry at University of Wisconsin at Madison with Professor Howard Whitlock. That was followed by a California President's postdoctoral fellow in Professor Larry Overman's lab at UC Irvine before joining faculty at Montana State University around 20 years ago. She served as a department head in her department from 2012 to 2018, and she's received a number of awards, including the NSF Career Award and more recently Montana State University Honors College Distinguished Faculty Award. So I will hand it over to Professor Cloninger for the lecture.
Thank you, Matt, for that introduction, and thank you for the opportunity to speak today. What I'm going to tell you about today is work that the bulk of that work is captured in this publication that's shown down at the bottom of the title slide there, in case any of you need more details or would like to see it later. And I'll start by acknowledging the people who did the work. The graduate students who completed this work are recent graduates of the lab, Sam Bernhard and Mackenzie Fricke, and then to thank the other graduate students and undergraduates in the lab. The collaborator on this project was Rainer Haag at Free University in Berlin, and funding sources that have supported this project, including the most recent funding source shown at the top there. So as said in the introduction, one of the important things to understand in this project and to realize in this project is that there are many times when a single ligand binding into a single receptor binding site is a weak interaction that may be too weak to be physiologically relevant. And the way that nature overcomes this is to have multiple ligands binding to multiple receptors synergistically, and that's the multivalent interaction, and that's actually a common motif when you have carbohydrates binding into protein binding sites. And there are a variety of biological recognition processes that are governed by multivalent protein carbohydrate interactions, and we've been fascinated by those interactions for quite some time. So if you like to study multivalent interactions, then frameworks on which you can display ligands become quite important. And in the work that I'm going to show you today, the bulk of this work is focused on dendritic polyglycerols. These are hyperbranched frameworks that are shown schematically here. And the appeal of these is that there's a high density of end groups that are there. So you can see over here, the ones that you'll see today are the 2.5 kilodalton, the 5 kilodalton, and the 10 kilodalton dPG frameworks, where you'd have about 20 hydroxyl groups on the littlest one, the smallest molecular weight one, about 40 on the middle-sized, and then about 80 hydroxyl groups on the largest dendritic polyglycerol. We abbreviate those frequently as dPG so that you don't have to say the whole thing. And then once you have the framework, in order to study the multivalent interaction, what I'm showing in this slide is how we appended ligands onto the dPG. So we functionalized the dPG with propargyl groups so that we could do click chemistry copper-1 mediated chemistry with azides to form triazole linkages to put on the lactosides. And so for the rest of the talk, when we have lactose-functionalized dPGs, you'll see that written as LDPG. And the important thing here, again, is that there are 3 different molecular weight frameworks, and for solubility reasons, we were trying for about 50% functionalization. So we have about 10 lactosides on the littlest dPG, up to 30-ish on the largest dPG. We were also interested in looking at lower levels, trying to explore where is the threshold of the degree of functionalization that you need to have so that you can have specific binding to a receptor binding site. And so on this slide, I'm showing you that we used mixtures where we just used the ethoxy ethanol with an azide on the end to block several of the hydroxyl groups so that you could have lower functionalization with the lactosides. So anywhere from really hardly any up a little bit higher in functionalization with the lactosides. Okay, so I've shown you the framework. I've shown you the ligand is just lactose in this case, and then the receptor that I need to tell you about is galectin-3. And on this slide, I'm showing the carbohydrate recognition domain, the CRD, of galectin-3, and you can see that there's a shallow binding pocket where lactose is bound into the binding pocket in this model system. And in general, galectin-3 binds well to terminal galactosides. It's prone to oligomerization because there's another domain that is an intrinsically disordered domain. It's called the N-terminal domain. And very little is actually known about this domain. And so in the rest of the talk, I represent the part that's better understood, the carbohydrate recognition domain, as its Pac-Man head here, where the lactose goes into Pac-Man's mouth. And then the tail we just draw as the squiggle because it's quite important. We can't forget it, but there's not a lot known about it. And one of the things that is really thought to be important is that those N-terminal domains can come together and intertwine, and that causes then the multivalent display of the receptor binding sites for galectin-3. So each galectin-3 you can think of as a monomer, but functionally it'll be in an oligomeric state during many of its processes. And then the other thing that I need to tell you about is that galectin-3 is important because it's overexpressed in many cancers. So on the next slide here, I'm showing you that there are many processes that have been shown to have upregulation of galectin-3. And as cancer progresses in many of those stages, galectin-3 is upregulated. Very little is known about the function of what galectin-3 is doing during those processes. And so what we were really interested in is developing multivalent ligand displays that then could alter the presentation of galectin-3 because overwhelmingly then we and others really would propose that galectin-3 oligomerization is one of the key points that's important for how galectin-3 is functioning during cancer progression. So if we make a multivalent framework and we study binding and aggregation processes of galectin-3, then we should be able to study cancer progression processes and learn what the role of galectin-3 is. So here are the project goals for this project. We wanted to use lactose-functionalized dendritic polyglycerols to induce aggregation of galectin-3. And then we really needed to characterize those aggregates because we wanted to use those aggregates then in cell-based assays. And so then the second goal of the project was to evaluate the effectiveness of the lactose-functionalized dendritic polyglycerols in cancer aggregation assays to understand what galectin-3 is doing during cancer progression. So in order to study protein binding by the frameworks, we use dynamic light scattering. And so what's important here to note is that we always use the same concentration of galectin-3. We always use that in excess. And we decided to set what I think of as a swamping excess of galectin-3, just an excess, and then a slight excess of galectin-3. So of course, if you have a swamping excess, some of your galectin-3 will be bound to your framework when you add it in, but some of it will still be free. Once you have a significant excess, you will have more of the galectin-3s that are able to bind. And then when you have just a slight excess, most of your galectin-3 will be bound. And so we do these experiments and we measure the sizes of the aggregates using dynamic light scattering. And I have this slide just to remind me to tell you that basically what we're doing when we're measuring dynamic light scattering is we're just taking laser light and we're shining it through a solution that includes our aggregates. And when you shine laser light through a solution with aggregates, the laser light hits the aggregate and it's deflected. And the angle at which the light is deflected allows you to determine the translational diffusion coefficient for your molecules. That's a different number for different size particles. So effectively, you can determine the hydrodynamic radius. How much space does each aggregate take up in your solution? And so that's abbreviated as R subscript H. That's the hydrodynamic radius. That's how big is your particle. So the first thing we do is we measure the hydrodynamic radius of the polymers themselves. And what you see is that the one that you might think of as the smallest polymer, because it's the lowest molecular weight, actually has the highest hydrodynamic radius. And the one that you might think of as the largest polymer, because it has the highest molecular weight, actually occupies less space in solution. It has the smallest hydrodynamic radius. And so we have a variation in the sizes of the polymers as they occupy space in the solutions. We then take the lactose-functionalized dendritic polyglycerides and we add those to galectin-3. So on the y-axis here, what I need you to notice is that these are big numbers. The hydrodynamic radius of each particle is really small. The hydrodynamic radius of the galectin-3 is in a comparable size range to the LDPGs. So what we're seeing is an aggregation phenomenon where the polymer nucleates formation of big aggregates. And this is fascinating to me because you can add these compounds together over and over again, and the aggregates are large, but they're also surprisingly homogeneous. So every time you do this addition, you get aggregates that are in the same size regime. And I find this fundamentally fascinating because it's not really clear to me that there's anything in the fundamental rules of supramolecular chemistry and non-covalent interactions that says that this should be true, but it is. Other things to notice on this slide, if you just have a slight excess of galectin-3, then it really doesn't matter what size the framework is. You get approximately the same size aggregate. If you use a more significant excess of galectin-3, then the hydrodynamic radius of the LDPG really begins to dictate the size of the aggregate. So using a polymer that is smaller gives you a smaller aggregate, and using a polymer that is larger gives you a larger aggregate. And this is true at both the 6:1 and at what, again, I think of as the swamping excess. That now the hydrodynamic radius of the nucleating polymer really plays an important role in dictating the size of the aggregate that's formed. I showed you the synthesis. These, if you remember the first three that are shown up here, the 2.5, the 5, and the 10, those were the ones that all had roughly 50% lactose functionalization. I included this green bar, a second 10 kilodalton, because it fits in the series beautifully. So if you remember, this is the one that actually has about 35% functionalization, and it has the smaller hydrodynamic radius and goes into the series really nicely for specific binding. So some control experiments here. If we use those compounds that I showed you that have a lower amount of lactose functionalization, once we dip down below about a third in functionalization with lactosides, we really begin to lose the specificity. So the particles are larger, the y-axis here is bigger, and the dendritic polyglyceride without any lactosides at all in general looks quite similar to the dendritic polyglycerols with lesser lactosides. So you can get non-specific aggregation, but the error bars are larger. The standard deviation on the particle size is much larger when you don't have the carbohydrate binding to rely on. And really, the aggregation that you see is indistinguishable from the non-specific associations that occur with the unfunctionalized dPG. Here's some control experiments where if you take away the propensity of the system to have multivalent interactions, then you lose this formation of large aggregates. So now the y-axis is quite small on this slide, just a few nanometers instead. And over here, I'm actually showing you the size of the galectin-3 without any LDPG in there. And so what you see is that the aggregates that you form when you use lactose, which can bind specifically into the carbohydrate binding site, or when you add mannose, those aggregates are approximately the same, and they're really, really small compared to what happens when you have specific carbohydrate binding in a multivalent fashion. If you instead allow the framework to be multivalent with the ligand display, but you take away the multivalent aspect for galectin-3. So here we've truncated off the N-terminal domain and used only the carbohydrate recognition domain. What you see here is also really, really small, essentially no aggregation. So if you don't have multivalent ligand display and multivalent receptors, then we don't see nucleation of these large aggregates occurring. And here I've just put the intact galectin-3 experiment with the 10 kilodalton LDPG on the same y-axis with the CRD alone to really show you the difference that large homogeneous aggregates can form very nicely when you have the multivalent system and not at all when you have the monovalent system. And I think that the important takeaway message here is that there's a lot of research to begin to understand the carbohydrate recognition domain that's really going well and really very beautiful. And we can't forget this N-terminal domain that I'm still just drawing as like a child's scrawl, and that there's going to be a lot of the story that's really quite important with the N-terminal domain and with understanding the aggregation and the functions of the N-terminal domain. I wanted to show you quickly the relationship between the LDPG and glycodendrimers. We've done a lot of work over the years with dendrimers. So here I'm showing you the dendrimer framework. Dendrimer is a Greek word that means highly branched or tree-like. And here the dendrimer is a macromolecule. So it's a more ordered system than the dendritic polyglycerol. Instead of a hyperbranched polymer, you have very systematic doubling of the number of end groups as you increase the generation number. And so what I'm showing you here is that we made lactose-functionalized dendrimers, and the generation 2, which has a theoretical 16 amino end groups. We have 12 lactosides on the outside of that. And so we made a series of G2, G3, G4, and G6 with 12 to 130 lactosides. And then over here, you can see the hydrodynamic radius. What you notice here is that unlike the dPGs, the dendrimers, as you increase the molecular weight of the compound, you increase the hydrodynamic radius. So in the dendrimer case, the compound, the G6, that has the largest molecular weight also has the largest hydrodynamic radius. And when you ask what happens when you add the glycodendrimers to galectin-3, here what you see is that, again, we had a small excess, a significant excess, and a swamping excess of the galectin-3. And the results are very, very similar for the hyperbranched polymer and the dendrimer. that again, when you have a very slight excess, the size of the polymer that's nucleating formation of the aggregate really doesn't matter as much. When you have a more significant excess, now you make larger aggregates, and the size of the nucleating polymer really plays a significant role in the size of the aggregate that is obtained. And if I just for a sec put these side by side, what you notice is a striking similarity in the shape of the charts, that the trends for the LTPGs and the glycodendrimers are very similar. And I think the takeaway message that I would give you here is that the hydrodynamic radius of the nucleating polymer plays a very specific role in making these large homogeneous aggregates. So then the next step is to take these and to put them into a system with cancer cells. And when you're putting them in with the cancer cells, here I'm showing you just cancer cells floating around in PBS. And I'm showing you that when you add galectin-3 into the cancer cells, then you see a significant aggregation. So this is a nice in vitro model of tumor formation. It's widely used as a model to mimic tumor formation in an in vitro setting. And so, of course, the question is, what happens when you put the glycopolymers into the system? And what we found in this case is that if you look at the y-axis here, if you just have the cells floating around by themselves, we've normalized that to 100%. And if you add galectin-3, then we have significantly less free cells. So a shorter bar means a lot of aggregation of the cells. And here I'm showing you if we add the 5 kilodalton lactose-functionalized dPG with approximately 20 lactosides as end groups, initially when you add the LDPG to the galectin-3 and the cancer cells, what happens is an inhibition of the galectin-3 mediated cellular aggregation. So at lower concentrations of the LDPG, you have inhibition and you begin to see free cells again. And then as you increase the concentration of the LDPG, what you see then is a synergistic effect where the LTPG and the galectin-3 work together to make more aggregates than we had before. So I would say that the LDPG appears to inhibit cancer cellular aggregation at low concentrations, but the LDPG enhances the galectin-3 mediated cellular aggregation at high concentrations. And if we compare that to the glycodendrimers, this graph is showing you glycodendrimers, how many free cells do you have. So here, the blue line is the generation 2 glycodendrimer with just 15 lactosides. So it's one of the little glycodendrimers. When that is added, if you follow the blue line across, what you see is that when you just add glycodendrimers to cells, nothing happens. If you add glycodendrimers and galectin-3, then the galectin-3 induces aggregation, but the glycodendrimer inhibits that. So you can affect cellular aggregation with the galectin-3, but then you can inhibit that when you add the glycodendrimer into the system. If we use a large generation dendrimer, here's a generation 6 dendrimer with 100 lactosides on the functionalization, what you see is exactly the opposite effect. So in this case now, you don't even have to add galectin-3. What you see is that as you add increasing concentrations of the glycodendrimer, you see cellular aggregation occurring in the system. So the cells begin to aggregate when the galectin-3 is added. And if you add galectin-3, you see maybe a slight ability at higher concentrations to overcome that. But overwhelmingly, what you see is that the large dendrimers with lots of sugars are able to induce aggregation, whereas the small dendrimers with just a few sugars are able to inhibit aggregation of the cells. And the reason that we chose the LDPGs that we did is because this LDPG in one way looks like the generation 2, and in another way looks like the generation G6. So the 5 kilodalton LDPG has a relatively low number of lactosides. And so in terms of the number of carbohydrate ligands that the LDPG is presenting, it looks more like a small generation dendrimer. And what we see is at low concentrations, it behaves like a small generation dendrimer. If we think about the generation 6 glycodendrimer, the 5 kilodalton LDPG actually has a hydrodynamic radius that's much more similar to the G6. So as far as the size of the nucleating polymer, now the 5 kilodalton and the G6 are comparable. The G6 has a lot more sugars than the 5. And what you see is that at high concentrations, the trends seem to follow the hydrodynamic radius, where now what we see is enhancement of aggregation at high concentrations that kind of matches what the large dendrimer is doing. And so what we could say is that now with the LDPG, the thing that's really interesting to me is that we have a framework that can inhibit at some concentrations, inhibit cellular aggregation, and then at other concentrations can enhance cellular aggregation, and that we can tie that to the ligand density and also to the size of the nucleating polymer that the galectin-3 is forming the aggregates around. So this is the last slide that I have, and what I want you to take away as take-home messages from this is that the lactose-functionalized dendritic polyglycerides, when they have a larger hydrodynamic radius, then the galectin-3 glycopolymer aggregates that will form will be large aggregates. When you have a smaller hydrodynamic radius for your LDPG, then the aggregates that form will be smaller aggregates. So you can really control the size of the aggregate that is formed by choosing a different size for your nucleating polymer. You have to have specific binding to the carbohydrate recognition domain. You need that protein-carbohydrate interaction in order for these large homogeneous aggregates to form. When you take away the specific interaction, then you still see aggregation, but it's much more heterogeneous. You might see large aggregates, as I've tried to show in cartoon form here. You see more heterogeneity in the sizes of the aggregates. The N-terminal domain is such an interesting part of the story, and still we have so much to learn. It's in our hands absolutely required for the glycopolymer aggregation to occur. When the N-terminal domain is removed, then we don't see significant aggregation. Importantly, you can use these compounds in cancer cell-based assays to begin to elucidate what is happening during different stages of cancer progression. And when we compare the LDPGs and the glycodendrimers, what we see is that the trends for the two significantly different polymer frameworks actually emulate each other quite nicely, and that you can take lessons learned from the one polymer system and apply them in a different polymer system and with predictable and understandable results. So I'll stop there and say thank you again for the invitation, and that I'd be more than happy to answer any questions that you might have.